My name is Coby Carlson. I work at CDI, and I'm giving this presentation, tutorial this afternoon, titled Driving Diabetes-Related Research Through Pathway Dissection of Human iPSC-Derived Cell Models. And this is kind of actually a continuation of a presentation that we did last year at SLAS in Washington, D.C., and it's kind of highlighting a three-way collaboration that we've got going on with BMG Labtech and Cisbio and CDI that has actually been really fun to continue working with them on these things. And so last year we had 3 different people come up and give detailed presentations about their own technology and the overview, which was very great. I decided this time to consolidate and put it into one presentation, one speaker. I'm not sure how I got voted out of the 3 of these people, but that's how it went. And and what I'm going to do is try to actually be light on the company overview stuff. And so they're just 3 slides on each of them. So I'm going to jump right into that and hopefully do it some justice. So I'll go ahead and I'll start kind of with CDI and giving again, a 1-slide overview of what we do and what we're about. And I'm not going to spend a lot of time talking about iPSC technology. Hopefully I assume that everyone has a basic understanding of that. But really, the power of this and why, we think and others think that it's, has, a high potential, a lot of promise in the field is that, you can take, the idea is that you can take a sample from any person, any donor across the world, any individual, that from there you can reprogram their cells back in time to a stem cell, to an iPSC, which as you can see here, I've actually got a pointer, you don't need to walk through it, but we've got reprogramming, we've got iPS cell generation, and at that stage you can do lots of different things in terms of engineering, and there's, gene editing technologies, CRISPR, TALENs, et cetera, where we can, change what we need to at the iPS stage, but then after that, going on and differentiating into many different terminally differentiated cell types. And you can see a lot of those listed, and under where it says iCell and MyCell, our iCell products are our normals, are from one single donor. And all of these that are catalog items on there, cardiomyocytes, skeletal myoblasts, et cetera, they all come from a normal healthy donor. And what, of course, the transformative power and the potential of iPSC is to get into the different donors. And so, like I said, not going to spend too much time on it, but many people know how to reprogram and do differentiation. I think what has made CDI different in terms of industrializing this is that we just do it at a really large scale, and there's some numbers about how we manufacture cells. And I think that's one key thing. Also, it's really important to be able to use cryopreserved cells. I think that's kind of an underappreciated fact that it offers you flexibility in terms of planning, when you want to run an experiment, how many plates you need to use. It's a really unique feature for this kind of biology. And then, of course, we just have done this for a long time, and we do it well, and so consistency is always a real challenge. It's not to say that it's always perfect, but we do as good of a job as we can to make it work out right. So that's iPSC and CDI, and so I work in a group that is focused on showing how you use these cells. And so we run a lot of assays, and we need to use lots of different tools and technologies to do it, but a lot of our stuff is plate reader-based assays. And BMG Labtech offers many different options for this. We use the ClarioStar, and it's a multi-mode plate reader. I'm just going to click through some of the things here and the highlights and kind of get rid of the animation. But the point is, this is a monochromator-based instrument that really has some unique features that allow you to, tune the wavelength that you're looking at, really, down to a very precise number, exactly what you want. And very sensitive instrument with different adjustable dichroic mirrors. You can read from the top, you can read from the bottom, you can focus right in on the cells at the bottom of your plate, and of course you can then do not only monochromator-based experiments, but there's spectrophotometry and luminescence, and you can see it's multi-mode in one with all these different technologies and endpoint TRF, importantly, multiple different well types. Down to 1536. It's got a dedicated xenon flash lamp, great PMT, which offers a really highly sensitive readout. superb dynamic range for the luminescence assays, and again, like I said, important to show that it's HTRF certified. And that leads us into the next, so this is BMG and their plate reader. I'll talk a little bit more about some things at the end, but this is the one slide overview for the Clariostar, and that gets us into Cisbio. And I think that there are a lot of different ways to detect cell signaling events. And there are a lot of pluses and minuses to a lot of different things, but traditionally people are relying on an ELISA and the ability to take an immuno-based assay, 2 antibodies, and miniaturize it and make it high throughput. Cisbio does a really good job of that. And so they've kind of redefined how you can run these cell-based TRF assays. And starting at a biochemical level, probably at the beginning, then working on a lot of the data with recombinant cell lines and overexpressed systems, which are very valuable and powerful by their own right. But now getting into what is considered more physiologically relevant cell-based systems, whether it's primary cells or stem cell-based or 3D models, however it is. And they've got an extensive catalog that really is superbly covered a broad range of things in terms of kinases and GPCRs, different biomarkers, and getting, of course, now into epigenetics. And it's just a pretty straightforward, ready-to-use, out-of-the-box solution with, in general, good supporting data to validate what they're trying to show. And just a schematic on the bottom where anyone that's run a cell-based assay where you've got your cells in the plate, you stimulate them, you lyse them, and you detect. And it's homogeneous and it's robust, so we're really happy with this, and it's a fun technology to use when you're trying to answer some of these different questions. So 3 things together, does it work? Well, like I said, we did this last year. We had some previous success showing this, so I'm just putting this together on one slide where here's an example of putting these 3 technologies and companies together to generate, I think, some pretty interesting data. So I'll go over this again. I presented this last year, but what this is showing an iPSC-derived Alzheimer's model. And so this is taking our normal IPS line and engineering that at the IPS level to introduce 2 different mutations. And they're listed in there. They're 2, disease-relevant mutations that modify the amyloid precursor protein, and they're actually at the same amino acid site, 673, and an alanine mutation. And one change makes the disease-relevant modification, makes it protective. And again, there's a lot more to talk about. And then the other one makes it pro-amyloidogenic. So it's really kind of interesting. We can make 3 different IPS lines from those 3 lines. We differentiate to neurons using the exact same protocol, and at the end of that, testing these 3 different neurons in a dish, we try to quantify the amount of A-beta protein that was released, peptide that is released. So, the mutations that are introduced for this APP modify the processing of amyloid precursor protein, and then the fragments, the peptides that come out of it. So what you can see here is that in the gray bar, this is the wild type, so this is the normal control. And you look at the protective mutation, which is the A673T. This is in blue. This is lower level. This is Y. On the y-axis is the concentration of A-beta protein, or peptide that is detected. And the pro-amyloidogenic, one that should make more A-beta, is actually featured here in red, A673V, and that has higher levels of the peptide detected. So this was actually a good illustration of how the genotype that was engineered, it really represents, in the biomarker phenotype. And it's always nice that, someone else generates data similar to you. So this is data from a customer, Genentech, that bought and used the same products and ended up, coming up with the same answers. So here's the data they published. And so this is, I guess, good evidence that this can be successful. The concept of this genotype to phenotype. So we've now built on this. What we're trying to do is, as we develop these assays in our applications group, we want to show people how they work and being able to translate that and get it out. So again, this is a simple overview slide of how we've done this. I'm not going to go into all of the details here, but the point is that we like to try to make it simple and flexible and amenable to a lot of different things. So if you thaw your cells and put them in a 96-well plate, you want to be able to take a look and see what those cells look like in the plate, because the assays take several days to happen. And so it's convenient, like I said, cryopreserved, thaw and go into 96 weld, look at the cells, and then once you're ready to run the experiment, they can be stimulated, and you can do multiple conditions across a multi-well plate. And then once the assay is done, you lyse the cells, and then from there you can put them in lyse them in a volume that's sizable enough to then separate it and put it into different readouts. So if you want to look at 2 different phosphoproteins, you want to look at an acetylation event versus a phospho, you want to look at total protein and phospho. So there's a lot of, again, flexibility to that. And then you can read this out on the ClarioStar plate reader at the end, which again, they've done a good job of coming up with defined settings, and all the stuff is really try to take the thinking out of it for you to be successful. So that's the protocol, and I wanted to spend one slide here also on the analysis of the data. And BMG also, I don't have this featured here, but they have a whole data analysis software package that helps do these things for you. This is just Excel and Prism based analysis. But what I want to show real quick that is, in this 2 antibody approach for an immunoassay, you excite at 1 wavelength and you read off 2 different wavelengths. It's ratiometric HTRF assay. And there's a lot of different processing of the data after it's done, but there's an excitation wavelength, then you read 2 different wavelengths, and you take an acceptor emission and divide it by the donor emission to create what is called an HTRF ratio. And in some instances, you can multiply this by 10,000, and you see data looks like this. So what you're seeing here is, and this is the plotted values of the excited, of the donor, of the acceptor emission. This is the donor emission, and it stays flat. So this is how you create your HTRF ratios, and then you can do what is processed and their data reduction as a delta F percentage. And then you can get fold changes each of these ways. So again, I'm just showing you all these examples here because admittedly the rest of this presentation has lots of different things. You might see different things on the y-axis, but the point is that all of this comes from a ratiometric HTRF readout when you're using their data. So perfect. Okay, so now this sort of gets into the story here, and again, why we are using iPSC technology, and what people are trying to do with this. And in general, one of the main things you hear about is disease modeling. And so this slide illustrates really kind of 3 classes of modeling disease in a dish, and we have them listed here as induced, innate, and engineered. And so you can see here that an induced model would be that you have a normal, healthy donor, cells are reprogrammed back to the iPSC state, and then you could put the cells under conditions of healthy, normal state, or disease modifying or disease inducing. And then you've got your terminally differentiated cells, and you can run an assay on those, phenotype assay, functional analysis, whatever it is. So induced, that's how this goes from one donor. And innate is really what people are very interested in. This is access to the diversity of populations, but you can have now here a healthy donor and a donor with the disease. And you reprogram those with the same method, you get iPSCs, then you go ahead and differentiate those, and you make the same cell type, but one looks normal and one is not. One is the mutant line, and then you can do the assays to compare these. All right, and so then the third one would be engineered, and I described this a little bit in the previous example with Alzheimer's, but this is where you've got the healthy donor, and differentiate to the, reprogram to the iPSC level, engineer in the mutation that you're looking at and you're interested in there, and then do those, you have now an isogenic control, do these 2 comparisons where you differentiate and test those. So really 3 powerful ways to do it. All of them have their, pros and cons definitely, but they are, the induced model is actually maybe the easiest one to start with in some instances, and so those are some of the examples that we'll talk about here today. And so the disease, again, that we're going to focus on is talking about diabetes. And one of the important features of diabetes is insulin resistance. So I'll just take a minute to talk about a couple things on this slide and set up the next few parts of the talk. So I've got the schematic on the left, the cartoon that shows the relevant cell types or tissue types that are important in diabetes. And if you are familiar with how this works, really, there are, to oversimplify it, some hormones, insulin and glucagon, that are involved in regulating, how these different organs handle glucose, how they take up glucose and how they make glucose. And again, that's really the simple way to do it. And the problem in diabetes is that those organs get desensitized and they don't respond to the hormones, specifically insulin, like they should. So that's what insulin resistance is. It's a reduced or attenuated response when insulin is around. And it's a characteristic feature of this disease, plays a big role in the pathogenesis of this. And to understand a little bit more about it from not just the physiological level, but talking about leading into the next part of the talk, the PI3K AKT pathway is really important. And it's essential for normal glucose transport. So, like I said, to understand diabetes, we want to understand how the cells normally process and handle glucose. And iPSC technology gets you access to these human cell types to really study the relevance of this. And the two cell types that we're going to focus on today are from the liver, hepatocytes, and skeletal muscle. And the liver, of course, is one of the main things it does is it produces glucose. And skeletal muscle is an important target tissue to take up glucose. So that should set the stage for the next little bit here. All right, so we'll start with hepatocytes first. And again, I try to sometimes oversimplify there's a minimum thing that a new cell type should do. An iPSC-derived cell type, if it's a cardiomyocyte, it should probably beat in the dish. That's what minimally you want it to do. If it's a neuron, it should probably form networks and form synapses and fire electrically and be able to communicate that way. If you're looking at hepatocytes, I think that you should know how it produces glucose. That's something it should do. So this slide just shows that, as you can read in the title, insulin decreases glucose production in hepatocytes. So the figure over there just shows sort of workflow arrows. Insulin comes in and decreases gluconeogenesis. And again, we'll talk a little bit about what glucagon does, but glucagon, another hormone that stimulates the formation of glucose, the production of glucose. So what you're seeing here, images of our iPSC-derived hepatocytes after a week in culture. Here they are just in a bright field and look very hepatocyte-like, standard morphology. Here's an image of just staining of the cells with some fluorescent markers at the same time. And over here is data from a gluconeogenesis assay where the cells were treated actually they were loaded with a substrate that would be used to make glucose, like lactate or pyruvate, and given a period of time and then stimulated with insulin. And you can see that, here's a dose response curve, here's the concentration of glucose on the y-axis, and that at the low concentrations of insulin, the cells are still able to produce glucose. But as you stimulate with higher concentrations, the amount of glucose that's produced goes down. And so an important part of this, although, like I said, we're talking about HTRF assays on here, we have to show functionality in a lot of different ways, and we think that the HTRF format is really complementary to what we want to do, and that's what we're trying to help show. But this is just a standard luminescence assay in this case, and again, underscoring how the Clariostar can be used for a lot of different endpoint readouts. Okay, so we are going into now talking a little bit how you can modulate insulin signaling, and I cell hepatocytes. So you've got a schematic of a pathway diagram over on the left that you may or may not be familiar with. The point is that insulin binds the cells on the outside of the cell, transmits a message, and you get a signal cascade down there. And you can see some of the important players in there downstream of AKT, and in this we're showing just all the glucose-related functionality that comes after a response and stimulation with insulin. So what we have here is just data to show that in a dose response format, you can treat the cells with insulin and you see an increased phospho event on the top, and in the bottom, in the presence of insulin, you put an inhibitor of the pathway and you can see a decrease in that cell signaling pathway. And again, not going into the details or the specifics of the pharmacology, but the point is you can modulate this signal up and down. And we've been collaborating with Cisbio, and they've got a group that does similar things application-wise to us, and transferred this over to them, and they ran, using our cells, generated some similar data. And not only did they, just look at phospho-AKT, but they expanded it and looking at total at the same time. And then they started evaluating some of the other downstream players in this and showcasing that those can also be phosphorylated in response to insulin. So the point is that the pathways are intact, and giving relevance to this model and understanding how insulin signaling goes. Oh yeah, and there's a poster on this. Some of the Cisbio people are presenting. I think it's actually probably today, could be right now. So you don't have to leave to go see it, but you could wait. That's fine with me. All right, so here we go. Glucagon-mediated cell signaling events. Again, sort of the opposite of response to insulin that you see here on the left-hand side. We have a different cell signaling pathway, but complementary to this glucagon basically just has the opposite effects. So what you can see here is that you've got glucagon binding, a glucagon receptor, and different things happen downstream. Cyclic AMP is produced, CREB is phosphorylated, and that can transcribe the message to go say, start making more glucose to the hepatocytes. And so here's some data on this, just sort of roughly addressing cyclic AMP here with some known agonists like glucagon and other ones related to this that elicit a cyclic AMP response. Here's a phospho-CREB assay, like I said, downstream at this point, and then similar to the gluconeogenesis assay we just showed on there, this is just glucagon-induced formation or production of glucose. So again, using these tools to slice up the pathways and making sure you know what's going on inside the cells. So then once you have an understanding of some of those events, it's also probably worthwhile to think about stressing the cells and trying to induce a disease condition like we were talking about. And to do that, you have to kind of know what pathways are active, just like we were doing for insulin. So here, showing some pro-inflammatory cytokine signaling in cell hepatocytes, where all of these are pretty much basal versus treated, where you've got IL-6 to induce a phosphostat-3 response, TNF-alpha, IL-1 beta, the combination of those to look at P38 MAP kinase phosphorylation or c-Jun phosphorylation or NF-kappa B. So again, pretty straightforward data, not dose responsive in this case, just using stimulating concentrations of the cytokines, but really reinforcing the fact that the pro-inflammatory response is there and cytokine signaling is intact in these cells. Okay, so I'm going to switch now from hepatocytes and go to skeletal myoblast and give a little bit more of the story here. So for skeletal muscle, one of the challenges is that the primary source is in general rodent cells. Of course, you can get primary human tissue, but the traditional models that people are using are C2, C12s and L6 myotubes. And definitely work. There's a lot of precedence with these, but there's variability in the differentiation from a myoblast to a myotube, and that can have just a different response. And so, of course, getting into the idea of disease relevance and human relevance, we generated, iPSC-derived skeletal myoblasts, which overcome a lot of these hurdles in terms of getting from myoblast to myotubes in a short time window, 3 days, with very consistent and high efficiency differentiation to the myotubes. So this is the introduction to skeletal myoblasts, and just like I talked to you about, if the cardiomyocytes beat, that's what they need to do. Hepatocytes they make glucose. Skeletal myoblasts should take up glucose. That's really one of their primary roles, is to take up glucose in response to insulin. And that's exactly what they do, and that's what this slide shows right here. So on the left, you're seeing just an ICC stained image of the cells at day 3 in culture after they've gone from the myoblast to the myotubes. And this is now data, again, another bioluminescent assay that something we've worked on with Promega in town in Madison, Wisconsin for a little while now, but this has turned out to be a really nice, reliable assay. And so I'm showing, again, raw data where you can see under basal conditions there's some glucose present, and you stimulate it with insulin and you can get a nice increase in glucose that's detected, and you put in a known inhibitor that blocks this uptake from happening, and that decreases below the case. And so in general, we're getting twofold or more changes once you normalize this data. This is cyto B subtracted data, and it's dose responsive, and it works really well. So this suggests that this cell model can be used for stimulating glucose uptake. Again, it also underscores the multifunctionality of these assays were run on the Clariostar. Okay, so you can kind of see the pattern of this. just showcasing some of the ways to dissect the cell signaling events, and again, very similarly, using AKT phosphorylation assays, looking at dose response of insulin at the same day 3 in culture. With a nice fold change in the window, you can inhibit this same response, the different phosphocytes for AKT with known inhibitors of this pathway, and then again some of these same cytokine stimulating events happening here, where you've got phospho-STAT3 responding in presence of IL-6 like it should, and P38 MAP kinase, in response to IL-1 beta, and et cetera. So this is, again, a similar pattern, just showcasing how you can take advantage of all the different content that Cisbio offers to, dissect your favorite cell signaling pathway of choice. Okay, so this was all kind of leading up to thinking about how to model disease, like we talked about, and it would be awesome if there were, some pathway that was something where we could engineer and we know that you make a mutation in the insulin receptor or IRS1 or something like that, and then we could make a complementary isogenic control and get these. Like I said, that's a little bit challenging. Identifying people with insulin resistance, there's a multifaceted disease. That's something we're still working on, but again, one of the first ways, sorry, I should have been clicking through this, the innate model, that's from the 2 different people. Engineering them would be the other way. But this would be an example of doing an induced system, healthy donor, and putting the cells under diabetic conditions. And it pulled off this silly image, but the idea is that you can't just feed the cells cheeseburgers and donuts, right? But if you want to try to examine how a high sugar and a high fat diet would impact how the cells signal and how they modulate glucose, that would be what you were trying to do, to induce the disease model of insulin resistance here. And so some of the causes, potential causes for insulin resistance, yeah, sure, you've got high concentrations of plasma glucose, you've got elevated free fatty acids, some stresses and reactive oxygen species and pro-inflammatory cytokines, that's present, or even just chronic insulin exposure, right? Your pancreas is working overtime. And so there's a lot of different things that can do this. So we were trying to, put this stuff together and see, well, what could happen? And again, this is just really the beginnings of doing this. But so this data here is using the skeletal myoblasts differentiated to the myotubes. And in that time period, and we decided to culture those in the presence of some of these different pro-inflammatory cytokines to see what the effects were. So this wasn't really an optimized set of conditions with, maybe the perfect amount of IL-1 beta, or maybe a combination of the two, but just what do each of them do on their own. And so this is kind of showing how you can do this. You've got a cell-based assay, you've got a technology that can read out something that is surrogate of glucose signaling at phospho-AKT, a high throughput, straightaway, easy assay to run. And what you can see here that is, really the control is in black, and there's not a great impact of IL-1 beta or IL-6 or TNF-alpha really on the EC50 of activation of this pathway. Yeah, there sure could be some things in terms of elevating the background or whatever that might be something that could be interesting, but of course, maybe not surprising. It was kind of nice to see that, this sort of chronic exposure to insulin, decreased the sensitivity both by the amount that was fold change in the assay, but also a little bit right shifted. I don't have the EC50 values on here. I don't know why I must have cut it off, but it's a, I don't know, 5-fold shift or something like that to the right, which is okay. So, and what's actually nice I thought about this was that, it didn't just cause an increase in background. Really, the lower level of phosphorylation was similar. So treating the cells with insulin had some effect on this insulin resistance. The next part of the assay was another way to do the same thing, and this was kind of, again, good data. I thought that, if you put the cells under conditions of oxidative stress, that also caused some problems with the cells and how they respond. And so what you're seeing here is that in red, there's the control curve with insulin, and then if you culture the cells in the presence of peroxide, this is actually just at a medium-ish concentration for about 2 hours. This also is another reagent that generates reactive oxygen species. Those two things caused a right shift of the cells in their response to insulin. So if you are becoming more insensitive or resistant, you would expect that it would take more concentration of insulin to give you the same response, and that's kind of what is being showed here. So anyway, like I said, this is kind of the beginning of this stuff. And after I put together this whole talk, I think, gosh, it would be really awesome if we had, the innate or engineered model to just jump right into. But I guess since we did this last year and we generated some new data for this year, I guess that'll just be what we talk about next year, right? So anyway, yeah, perfect, exactly. So anyway, so the summary of this is, I think HTRF technology is very powerful, well established in the community. People are very familiar with this and appreciate the content that is offered in there. Clariostar plate reader and BMG Labtech has been a great company to work with. They're really actually supportive of and understand how their instrument works. It does a lot of different things, and I mentioned about the data analysis. We don't even scratch the surface for the capabilities that it offers, so, but it gives us what we want to know every time, so we're happy with that. And iPSC technology, I think it's fair to say it's changing the way people are studying human disease, and hepatocytes and skeletal muscle are clearly important cell types to study diabetes. And I think a message from these kind of things is that, you have to have good cells and a good assay and a good way to read this out to understand if what you're doing makes sense. And so you have to get good quality data that way if you want to make data-driven decisions. And I think these things together have pretty much helped us to do some of that. So, and again, the idea for what we do and our group is we want to try to develop applications with normal cells and then be able to plug in the mutant ones and see the comparisons. I think it's a really simple thing, but the comparison of this induced, innate, and engineered disease modeling approach is really, I think, a good way to do this. So, all right, so before the last, I don't know, I probably spoke pretty quickly because I tend to do that, but I wanted to, since there's a minute, kind of link two things together, and it was just sort of a timely publication that came out. And since I talked about diabetes in here, but I also linked in a little bit to Alzheimer's. So this is a new paper that just came out from Stuart Lipton's group that it's titled Elevated Glucose and Oligomeric Beta Amyloid Disrupt Synapses via Common Pathway of Aberrant Protein S Nitrosylation. And so this was just, this is a Nature Communications paper that just came out in the beginning of this month. And so anyway, what they showed in this paper was that if you culture cells, neurons specifically, in the presence of high glucose or in the presence of increased beta amyloid peptide, that they were able to identify certain proteins that had disrupted or aberrant protein nitrosylation. And so what you're seeing here is this data from the paper. I think these are rat cortical neurons in this example. They also had human iPSC-derived. It's a really nice, thorough thing, but they showed, these IDE or DRP1, these proteins, and they can look at, nitrosylated versions of these proteins with high glucose and oligomeric beta amyloid. So what happens here is that, under these disease-induced conditions, once you have these modifications to the proteins, that renders them non-functional, and signaling goes bad, and that's what causes diabetes, and that what ends up causing Alzheimer's. So it was an interesting overlapping thing, and people have known for a long time the relationship between this, between all people that have Alzheimer's and their susceptibility for diabetes, and even the other way around. So. And what I thought was neat about that is that, Cisbio, although I was saying that they've got a lot of different coverage and content offered, they also have a way to offer custom versions of this. So you could envision that, hey, just like I showed you on this previous slide, I have an antibody to insulin-degrading enzyme, IDE, and I have an antibody that detects modified nitrosylated proteins. I want you to build me a custom assay that detects those two things together. And that's a solution that they offer. And our group at CDI, our applications team is really applications and support, and it's really focused on helping people understand how to use the cells and how to run them. So I think that this sort of custom offering and people that have a lot of experience with the cells could help direct in that way. So anyway, it was just sort of a food for thought type of thing, timely in the literature that I wanted to put out there. So anyway, and I thank everybody for coming. This is obviously the three companies were involved in this. These are the booths, and that's all I got to take questions. Thanks a lot.